Hi guys, welcome to my video CRISPR-Cas9 Gene Editing System. Um, you finally made it to week 13. You're almost done. This week's material is fascinating, and it's actually on the cutting edge of research here. This CRISPR-Cas9 gene editing system that we're going to talk about here this week was only discovered in 2012. And the inventors just got the Nobel Prize in 2020. So this is really, really cutting edge new stuff that we are just learning how to use. And it's really, really cool. So I'm excited to share it with you guys and how it works. Let's see if we can help you out with this. So in week 10, we learned how to insert a gene into bacteria. We used restriction enzymes to cut plasmids, glue the gene into the plasmid with ligase, and then insert the plasmid into the bacteria using transformation. In week 12, we learned that that process doesn't entirely work on higher order cells, like plants and animals. And so then we had to learn new methods like the gene gun and agrobacterium and viral transduction to try and get the gene into the cell so that we could glue it into the chromosome. Well, the tricky part then is now we need to talk about how do I get it into the chromosome? Because remember in week 12, we learned you have to actually get it into the chromosome in order to make it work on a higher order cell. So we can't just put it in on a plasmid. We have to actually glue it into the chromosome in addition to getting it inside the cell. Now with plants and some animal cells, we've got less reliable methods that that can insert the gene into a cell maybe. But if I'm trying to use this for therapeutic healing on a human, if I'm trying to like cure a human of hemophilia by, viral transduction can drop the gene off inside the cell. But there are times where I want to very specifically control where I put that gene. And the methods, for example, if I'm trying to replace just part of a gene, because some viral vectors are too small to hold the entire gene. So sometimes I want to replace just part of a gene. So I need to control precisely where we cut and precisely where we glue. Otherwise, it's going to be a no-go. Now, we learned in week 10 that you've got restriction enzymes. If you recall, restriction enzymes were enzymes that were capable of cutting the DNA at a specific spot. Restriction enzymes. They could cut DNA at a specific spot, and it was a palindrome. Right, so we had, like, for example, EcoR1 was a restriction enzyme that could cut at the palindrome GAATTC. We used that one a ton in the homework, right? And it chopped between the G and the A. This worked great on a plasmid because plasmids are only, a couple thousand base pairs long at the most. And so you'll find 2 or 3 EcoR1 restriction sites there, and you'll chop it in those few spots, glue your gene into the plasmid, and we're good to go. The problem is with an animal cell, and especially a human cell, you can't remove the DNA, work on it, and then put it back in. So we've got to work on this inside a living cell. The second problem is rather than having 1,000 base pairs like a plasmid has, humans have 3 billion base pairs of DNA. So what are the odds? You're only going to find 1, 2, or 3 cut sites for EcoR1, 1, 2, or 3 GAATTCs in 3 billion base pairs of DNA. That's just outrageous, right? There's going to be tons of them all over the place. So if I threw EcoR1 inside a human cell, it would shred the chromosomes to pieces. So not only would I cut the chromosome and try and glue the gene in, I'd cut the chromosome I'd butcher it entirely, and that would kill the cell real quick. So EcoR1, despite its fantastic ability to look for just one particular restriction sequence, is not specific enough. 6 base pairs is not big enough of a cut site in order to use on a human cell. There's just too much DNA in play. So we need something more specific than the restriction enzymes we learned about in week 10 in order to work on a human cell. And that's what the CRISPR-Cas9 system is. The CRISPR-Cas9 system was discovered in a bacteria called Streptococcus pyogenes. And it's kind of ironic because the bacteria, or because Streptococcus pyogenes can give you strep throat and a bunch of other nasty things. So it's a real nasty bacteria, and yet we find something extremely helpful on the inside of it. Turns out that bacteria can also get sick. Bacteria, there are viruses that attack bacteria and kill them. They're called bacteriophages. Anyway, so the bacteria has to have some sort of immune system to defend itself against the virus. And that's the CRISPR-Cas9 system. So CRISPR is an acronym. And you do need to know this acronym for the exam. So there's your note. Please know the acronym. CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. That's a really long acronym, hence why we use the abbreviated version, CRISPR. Clustered, regularly interspaced, short palindromic repeats. What this is this is basically a memory system in a bacteria. So every time they get invaded by a virus, they actually collect a bit of the virus's DNA and store it as like a memory thing to help the bacteria remember, well, this is what that virus's DNA looked like. And then they can actually recall that when they get invaded by the virus in the future and defend themselves. So their little memory mechanism is CRISPR. Hence why we carried the name over. So even though the CRISPR memory component isn't actually really part of our gene editing system at all, it's actually what was in the bacteria originally, not the gene editing system itself. We call it the CRISPR system because the original bacteria's system included the CRISPR component. So because the CRISPR component can kind of get a little confusing, I'm going to kind of glaze over that because we're more interested in the gene editing system, how this works for editing genes. But you should know the acronym, clustered, regularly interspaced, short palindromic repeats. So let's get to the other half of the name here, Cas9. Cas9 is a restriction enzyme, just like those ones that we learned about in week 10. It's a restriction enzyme. Which means it cuts DNA. But unlike the restriction enzymes we learned about in week 10, Cas9 does not have a memorized sequence that he targets. Instead, he can actually be told where to go. He can be programmed where to go. He can be programmed to look for a sequence up to 20 base pairs long, and it can be a very, very specific sequence. So this is really, really fantastic, especially in our realm of gene editing, because that means I can hijack this system and specifically program Cas9 to cut exactly where I want it to at a spot with a restriction site up to 20 base pairs long, which the odds of finding a specific 20 base pair sequence in 3 billion base pairs is a lot smaller than finding a specific 6 base pair sequence. Like the statistics on that are just crazy, right? So we can make this a lot more specific and actually program Cas9 to go exactly where we want it to go and chop just where we want it to cut inside the human chromosome to glue our gene in. That's really fantastic. So that's why this was such a huge breakthrough when they discovered this in 2012, is this allows us to get specific enough that we could actually use it to glue a gene into a human chromosome predictably or glue a gene or a part of a gene into a chromosome predictably, which is huge. So let me show you how this works. So Cas9 is this great big enzyme. I'm going to represent it by just this big circle, okay? This is Cas9. And here's the human DNA, double-stranded here, that I want to cut. So let's say we got Cas9 inside the cell. So we would drop it off using a viral vector here. So we're going to use transduction. We're going to drop Cas9 off inside the cell. Now Cas9's inside the cell along with the gene I'm trying to drop off, okay? So we've got Cas9 in the cell. So now here's the human DNA that I'm trying to cut inside the cell, okay? Cas9 can be programmed because he, rather than having a memorized sequence, he uses this little piece of RNA called a gRNA, which stands for guide RNA. And it's got this weird little shape that looks like this. This is our gRNA, which again stands for guide. And the reason why it's called guide RNA is this RNA tells Cas9 where to cut. So he basically guides Cas9 to the location where he's supposed to cut. That's the job of the gRNA. He's supposed to lead Cas9 to the cut spot. So we've got this little hairpin loop here. We call it a hairpin loop because it kind of, if you use your imagination, it kind of looks like a girl's hairpin or a bobby pin kind of thing. And then we've got this straight piece over here, and that makes up the gRNA. Well, wherever this little straight piece pairs up with the DNA, so this is the part that we can program because I can determine the exact sequence of this little straight piece of the gRNA. Wherever this sequence pairs up with the DNA, Cas9 chops. And Cas9, so if you remember in week 10, we always told you, pick a restriction enzyme that cuts with sticky ends. Cut with sticky ends, blunt ends are bad. Well, Cas9, ironically, cuts with blunt ends. Ha ha ha, joke's on you. Okay, so Cas9 cuts with blunt ends. And yes, blunt ends bad. It's not impossible to glue a gene in using blunt end cutters, but it's a lot harder than using a sticky end cutter, but we put up with it because Cas9 is so fantastic in every other respect that we can put up with the fact that it's a blunt end cutter. That's just kind of an ironic thing, is as hard as we pounded sticky ends into your head in week 10, now we're using one with blunt ends. But again, it's a lesser of two evils at this point because Cas9 is so fantastic everywhere else that cutting with blunt ends is a lesser evil. So wherever this little straight piece of the gRNA pairs up, it chops clean through, or the Cas9 chops clean through the DNA. And now we've got a cut. And we can either use that cut to deactivate a gene, like if I've got a gene that's doing something bad, I can use this to deactivate the gene, because when we got that cut, the cell will recognize the cut and try to fix it, but it will fix it badly, and it will actually ruin the whole gene when it tries to do that. So if we just want to deactivate a gene, then all I have to do is have Cas9 cut it and watch the cell ruin itself. Or I can use this to insert a gene, just like we did before. You insert a gene right there in that cut, and now there's the new gene or the new part of a gene that's in there. Now, it turns out that this gRNA, because we're scientists, we have to make things a little more complicated than this. So this gRNA actually has two sub-names. There are two parts to the gRNA. So it's actually one piece of RNA, hence why it's called the gRNA, but there are actually two names for different parts of gRNA, just like there are different states inside the United States or different countries in the world, right? They're all just parts of a whole. So this little piece that's, oops, I'm still on eraser here. There we go. So this part of the gRNA, notice it's just part of the gRNA, it's not a separate piece. This part of the gRNA, the part that actually pairs up with the DNA, is called the CR-RNA. That stands for CRISPR-RNA. CRISPR, again, because this part would have come from the CRISPR memory component in the bacteria. We just built it synthetically rather than having it come from the CRISPR memory component, but we still call it the CRISPR-RNA because of that. So the CR-RNA is the part that actually, the part of the gRNA that actually pairs up with the DNA sequence that we're trying to cut. This other part here is also part of the gRNA. So remember, the CR-RNA is still part of the gRNA. This other part is the little hairpin part right here. There we go. And this part right here is called the tracer RNA, TRACR, all lowercase, RNA. That actually stands for transacting transacting CRISPR RNA, but you don't need to know that or what that means. Just know it by the name tracer RNA and know this one by the name CR-RNA. So the tracer RNA, his whole job is for Cas9 to hold on to the gRNA. So he's kind of like the handle. That's his only job in life. Cas9 has to hold on to this assembly somehow. So he holds on to the tracer RNA half, and then the CR-RNA part is the business end. So here's kind of an analogy to help you keep track of all of these different pieces. Let's pretend that I'm at the grocery store. This isn't such a far-fetched scenario, knowing my memory abilities. Let's say I'm at the grocery store and I walk out, and not only can I not remember where I parked my car, which happens literally every single time, but I also can't remember what my car looks like. Thankfully, that hasn't happened yet. Let's say I can't remember what my car looks like, but I do have my car key. And so the most reasonable way, other than calling my wife and asking her to remind me what our car looks like, is to go and test my key on every car in the parking lot until I find the one that fits. So in my little analogy here, we've got the little keyhole on the car, right? And then we've got a key. Here's my little key. And then me holding onto the key here. Oh yeah, that definitely looks like my hand right there. Beauty, see? Good thing I'm not an art major. Okay, there's my hand right there, and I'm holding the key. I'm testing it out. And wherever the key fits and opens the door, that tells me, yep, that's my car. And I will open that door. So if in this analogy here, the keyhole, that would be kind of like the human DNA, right? The part that I'm testing on. The key, that's my guide RNA. So it's telling me where I'm supposed to open the door. And wherever he matches, that's where I cut. That's where I open the door. And then my hand is the Cas9. So he's guiding me, but so the guide RNA doesn't do the cutting. The Cas9 does the cutting. The key doesn't actually open the door. He just allows me to open the door. He tells me which door to open. So the key is kind of like my gRNA. And Cas9 is like the hand. Now a key, notice a key is a solid piece of metal. It's one whole chunk, but there are two parts to a key, right? There's this jagged part that you actually stick in the door, and there's this circle part that you hold on to. The jagged part is the business end of the key. That's the whole reason for having a key. It's the part that you actually stick in the door, and the part that's actually supposed to pair up with the lock inside the door. And if it matches, that's where I'm supposed to open the door. If the jagged part doesn't match, then I'm not supposed to open this door. So the jagged part of the key, the jagged is the CR-RNA. That's the part of the gRNA that actually pairs up with the DNA and tells me where to cut. This other part of the key, the round part out here, his sole job in existence is for me to hold on to the jagged part, right? That I'm supposed to hold on to that part so that I can stick the jagged part in the door and still hold on to the key and turn it. So that's the tracer RNA. Tracer RNA. So that's the, notice the gRNA is one solid piece of metal. Like the key is one solid piece of metal, but the jagged half of the key is the CRRNA, and then the handle part is the tracer RNA. So the whole goal of the tracer RNA is for Cas9 to hold on to the key. It's the handle. And then the CRRNA actually pairs up with the DNA and tells Cas9 where to cut. So by changing the sequence of the CRRNA, I can tell Cas9 exactly where to go cut. And that's the really cool thing about this system, and why it's so helpful. So that's the CRISPR-Cas9 system in a nutshell. It's once you've got it drawn out like this, it's actually not that bad. Just remembering all those different pieces. So Cas9 is the enzyme that actually cuts. The gRNA is the key, it's the guide, so that leads the Cas9 to where it's supposed to go. The CRRNA is the half of the gRNA that actually pairs up with the DNA and tells Cas9 cut here. And the tracer RNA is the half of the gRNA that Cas9 holds on to. And yeah, that's basically it. And this is a really, really powerful system. And we're still working out the kinks, but just 2 years ago or so, they did their first trials on humans, and they've actually cured some people of sickle cell disease. And a year later, they're still good. So they went in there and swapped out the bad version of their factor 8 or of their hemoglobin gene, and now they're cured of a genetic disease. And we're just now scratching the surface of what we can use on this, or of what we can use this for. So it's a really, really cool system. And I look forward to seeing what they do with it here in the future. So anyway, if you've got any other questions about the CRISPR-Cas9 system, please let me know. Reach out to me or your professor, and I'm happy to help in any way that I can. My email is dallin.stokes17@gmail.com. So reach out to me if there's anything I can do to help. And good luck to you guys.